Good afternoon. This is Dr. Dan Guerra coming to you from the lovely VerEvMed studios here in the Pacific Inland Northwest. Coming to you today on the 22nd of June, and we got a great presentation we're going to give you, and I want to get into it right away. So let's do that. From the beginning, like the Emerson, Lake and Palmer song. All right. Anyway, let's do a little bit of housekeeping. That's me. I'm Dr. Daniel John Guerra. I'm a lipid biochemist. I'm a former professor, semi-retired. I am also a research scientist, not yet retired, and I am a lecturer extraordinaire. What I'm doing today with my company VerEvMed is presenting to you yet another lecture on some key feature of the refereed biomedical, biochemical, cell physiological research journals. And so what I'm trying to do here is give you a topic that follows from the last several lectures. So if you're following along, which I know some of you do, you're going to see how this fits quite nicely. I'm going to also bring up, of course, diabontology, the new concept out there in biology. So without much further ado, I'm just going to say that this is the Facebook page you want to look at. It has all of the video lectures on it, my personal Facebook page, but also the company's from my vantage point. And so you're going to see some philosophy in there as well. So I do a lot of philosophy. I write in the area. And of course, my main practice is science. VerEvMed, there's our logo. Again, I'm Dan Guerra. That's my particular and personal email address. You can contact me directly about this video or any other ones you see, and I'd be glad to talk to you about the details of how I put this together, or perhaps consult with you on a potential new video. These are free. My company, of course, is a straight capitalist gain function where we try to gain clients and bring you into the scientific literature. And that's what we do for a living. So this is just an example of the kind of work we do, and that's why it's free. All right. So with that out of the way, the email address of the company is right there, and there's the website. Please visit us. Please become our client. We'd be glad to talk to you. The first visit, of course, the first discussion is absolutely free, just like this video is. Just because it's free doesn't mean it doesn't take a lot of time to put it together. And I think you'll be able to tell from the detail that that's clearly the case. All right, so what are we going to talk about today? How macrophage lipid metabolism is manipulated to overturn inflammation via a reversal of ligand receptor functionality. Did you follow all those twists and turns? I think you will once we unravel it. Okay, there's the paper we're going to talk about today. It's from an excellent journal called Nature Immunology. I had to wait a month for the embargo to come off of this paper. I did. I warned you I was going to do the paper, and here we are at it. Published this month, June. And let's take a look at some of the background information we need to know. Macrophages, which are, of course, our immune cells in the body, okay, typically classified as the innate part of the armamentarium of the innate immune response. They can be resident macrophages in tissues, and they can also be circulating. But another way to diversify macrophages is discuss them into whether or not they are inflammatory or pro-inflammatory or anti-inflammatory. And that's the topic of today's discussion. Now, inflammatory macrophages differentiate in response to microbial and tumor antigens, as you might guess. And in fact, interferon gamma is a good way to induce the inflammatory macrophage lineage, or polarity as it's called. What you do is produce pro-inflammatory cytokines at the site of nascent infection. That's what the activated macrophage will do. And it also occurs when things are working correctly at cancerous lesions, thus killing the lesion, thus killing the infection. And then, of course, promoting the further mobilization of other immune agents, such as the lymphocytes like T cells and B cells. But that's downstream, and that's not our topic today. Now, anti-inflammatory macrophages, as you might guess from the title or the name, differentiate via signaling by glucocorticoids, which of course are typically immune suppressive steroids, or sometimes by anti-inflammatory type 2 cytokines. What kind of cytokines are anti-inflammatory? Right there. Interleukin 4, 13, and 10. And they can actually induce a T cell H2 immunity lineage. And what Th2 immunity does, it starts to mediate tissue remodeling, wound healing, and modulates the immune response. That is, it tends to turn down the pro-inflammatory response. Now, IL-4 and 13 drive the anti-inflammatory macrophage polarization through the IL-4 receptor alpha chain, known as IL-4R alpha. And anti-inflammatory polarization is also promoted by the activation of several master regulators, including these: signal transducer and activator of transcription 6, or STAT6, Tolllike 4, factor 4, or KLF4, and the interferon regulatory factor 4, or IRF4. I'm not going to talk about those today, but I just want you to put together, if you've listened to previous lectures, or if you know something about the immune response, that's where they come into play. Now, let's take a look at the mediators we're going to be carefully looking at in this study. First of all, I want to say some background things. Bioenergetic reprogramming is associated with macrophage polarization. That is, the kind of fuel that the macrophage is going to use is going to help dictate the ultimate polarity of that macrophage. In this instance, the polarity is inflammatory or anti-inflammatory. Now, the inflammatory macrophage cell type is fueled by aerobic glycolysis, and it's triggered by lipopolysaccharide plus or minus interferon gamma. Okay, so your typical immune response, immune antigen induction, the inflammatory macrophage lineage is induced, it's activated, and the kind of fuel that's being used is glucose. We call it aerobic glycolysis because it works in the state of having molecular oxygen. So even though glycolysis is an anaerobic pathway, that is, it doesn't require oxygen, aerobic glycolysis is occurring in the presence of oxygen. That usually means the TCA cycle is functioning, active mitochondria, making a lot of ATP by utilizing the pyruvate at the end of the glycolytic pathway, which is cytosolic, of course. Now, with the anti-inflammatory lineage, IL-4 induces the expression of PPAR gamma. Now, this is a transcription factor we talked a lot about here at VerEvMed. Basically, it turns on beta oxidation of fatty acids. Now, that's really important because if you use fatty acids rather than glucose, you're changing the entire biofuel economy of the cell. Using fatty acids means you have to take them in. Of course, you have to take glucose in too with, say, GLUT4 transporter. With fatty acids, you need your own membrane receptors to take in fatty acids from circulating lipoproteins like LDL and VLDL. But you also need an entire nested framework for utilizing those fatty acids, which means binding to them in the cytosol, making acyl-CoAs, translocating them into the mitochondria via a carnitine pathway, and ultimately going through the beta oxidation to make acetyl-CoA. Okay, so PPAR gamma is responsible for turning on transcriptionally that whole system. It also transcriptionally activates urea cycle enzyme arginase, because during fatty acid utilization, you're also utilizing amino acids from limited proteolysis. Those amino acids have to be, the amino groups have to be converted or reassigned, otherwise free ammonium can be toxic. So the urea cycle is really important in terms of adaptation to fatty acid oxidation. It's something people don't normally talk about, but because I'm a biochem prof, it's what I always bring up. I want you to know that nothing happens in a vacuum. Fatty acid metabolism, yes, but also amino acid metabolism, particularly the urea cycle. And arginase is obviously an important enzyme in the urea cycle. So that's called ARG1. So you get ARG1 and the beta oxidation of fatty acids along with an uptick in electron transport chain, oxidative phosphorylation. That means you have increased mitochondrial biogenesis. Now, you also get increased mitochondrial biogenesis when you're anabolic. And we'll see why I bring that up later, because cell fate sometimes looks like it's collinear, but you can see where it diversifies downstream. Now, to fuel the anti-inflammatory bioenergetics, IL-4 also induces the expression of CD36. So now I'm introducing it. I've talked about it many times in previous VerEvMed lectures, and I'm sure some of you are fully aware of it. You can go back and listen to those. But anyways, it's a membrane receptor. It's also considered an orphan receptor, but it is able to bind circulating low-density lipoprotein and very low-density lipoprotein, extract the triacylglycerol and to some degree also the cholesterol from those lipoproteins. It takes the triacylglycerol and it utilizes that as a source of fatty acid. So finally, the unloading of the triacylglycerol, or TAG, we call it, an associated fatty acid hydrolase, or which is a lipase, lipoprotein lipase, is linked to then the beta oxidation, thus completing the anti-inflammatory polarization. Once all that goes down, all that biofuel change occurs, transition occurs, you're now full blown going into the polarity of anti-inflammation. And anti-inflammation is really important because if macrophages don't also turn down inflammation, so you have macrophages that are pro-inflammatory, and then you have the anti-inflammatory. If the anti-inflammatory aren't ready right following in a sequence from the pro-inflammatory, you can get full-blown chronic neuroimmune and total global immune responses, and that can cause a lot of tissue damage, cell damage, DNA damage. So you want to be able to, as soon as the immune response is kicked in, and you get an inflammatory response in particular, which is what macrophages essentially initialize, you want to be able to have a circuit directly to be able to turn that down. And that's how this occurs, this transition from pro-inflammatory to anti-inflammatory with one cell type utilizing different biofuels. That's the whole key to this polarity and keeping disease state at a minimum. This is particularly important in lots of autoimmune diseases and pro-inflammatory chronic diseases that are linked to, say, obesity. A very common pro-inflammatory disease, chronic pro-inflammatory disease, is all the ones associated with the gut. So, for example, inflammatory bowel disease. That is a disease that is linked to having a hyperactive immune response, which means there may be something wrong with this lineage switch to the macrophages, which are anti-inflammatory. In fact, that's ultimately why the study was done. Now, let's talk about some more players. These are all mediators. Again, we talked about MTOR, that is the molecular target of rapamycin. Rapamycin is an antibiotic that needed to have a home. Where does this antibiotic work? Well, it was determined the target of it was this transcription factor. Actually, much more than a transcription factor. It's a global switch in the cell. So within the other polar phenotype, pro-inflammatory, mTOR responds to energy demands, thus involving increases in glycolysis. So this is in the pro-inflammation mode. mTOR is functioning there. But also the anabolic program of lipogenesis and protein translation, because remember that mTOR is an anabolic system. So it's also involved in producing enough energy to go through cell division, cell cycle to cell division. Okay, so that's what mTOR is involved in. Protein synthesis, lipogenesis, which makes membranes, cholesterologenesis, which further makes up the composition of the membrane, and the sequence of events that causes the membrane to ultimately allow fission and allow, of course, cell replication, along with DNA synthesis, right? DNA replication and protein synthesis, as I just said. Now, mTOR activation has been linked to metabolic reprogramming in both macrophage polarization and in general in the innate immune cell activation system, okay? Now, mTORC1, don't worry about that. TORC, that just means complex. So now you have the target of rapamycin, that one protein, associated with lots of other proteins, and that's the big C, the complex. There are two mTORCs, 1 and 2. We'll see how they come together soon. At any rate, they promote cholesterol and fatty acid synthesis through a pathway involving actually tuning down PPAR gamma and the steroid regulatory element binding proteins 1 and 2. Normally, PPAR gamma and the SREBPs are involved in what? Well, those are steroid regulatory binding protein normally involved in cholesterologenesis and also in fatty acid synthesis, whereas PPAR gamma is working in the opposite direction, doing fatty acid breakdown via beta oxidation. So I'm just telling you that this is the pivoting point. This is the teeter-totter where the pivoting point is. Now, mTOR is going to be able to put its hand on both sides of that teeter-totter or that balance or scale. We'll see how. When active, mTOR triggers cell growth and proliferation by promoting protein synthesis, lipid biogenesis, and metabolism thereof, and by reducing autophagy. Remember, autophagy is the starvation mode, right? When the cell is going through autophagy, it's not anabolic. So whenever you seek out a cell that's autophagic, mTOR is going to be turned down or off, okay? Nothing is really ever off in the cell, but turned way down. Now, mTORC1 inhibition has been well studied because obviously mTOR activation can lead to cell proliferation, not only a pro-inflammatory response, but also what? Cancerous or precancerous growth leading to full-blown oncogenesis and tumorigenesis. So mTOR has been a target of study in biomedicine for many, many years, ever since its discovery. Now, mTOR inhibition, in addition to reducing protein synthesis, that is when you inhibit it, deeply affects gene transcription in general, tends to turn it down, okay? But differentially. Now, here's a good diagram showing you what I mean by putting it all together. You get a growth factor that stimulates this phosphoinositol 3 kinase that will take phosphoinositol bisphosphate and turn it into a trisphosphate right in the membrane. This is a membrane lipid, okay? P10 is just an enzyme which normally will block that phosphorylation of that phospholipid. Anyways, once you make PIP3, which again is inositol trisphosphate, you activate the protein kinase D, PDK1. PDK1 phosphorylates a tyrosine residue, shown there, on an enzyme called AKT. This is canonical mTOR pathway stuff. Once AKT is phosphorylated there, mTOR2, also working with a cofactor protein called Richter, it generates the mTOR2 complex. There's other proteins involved, actually. We're not sure what actually activates that. There are lots of things that co-stimulate. But you phosphorylate a serine residue. Once fully phosphorylated tyrosine serine, you then block this tuberous sclerosis complex. Now, the TSC complex would normally block mTOR. Okay, so if you block that, what happens is that you unblock everything downstream from it. Yeah. Now, look what normally turns on the tuberous sclerosis complex: AMP kinase. AMP kinase comes from glucose deprivation or oxygen deprivation. ATP goes down. AMP to ATP ratio goes up. AMP kinase goes up. TSC then, okay, gets activated, and that shuts down anabolism. We're not talking about that today. We're talking about activating this pathway. AKT blunts or shuts down or blockades TSC. So you're able then to carry on mTOR activation via the REBGTPase. Again, with Raptor as a cofactor protein. The mTOR1 complex fully on. There's where rapamycin would normally inhibit. And then it turns on these two bifurcating pathways, ultimately leading to anabolic pathways, cell growth and proliferation. I got this off my paper published in JASN 2009. This is where this figure came from. Just one of many things you can pick up online to show you the canonical mTOR pathway. Now, enter the semaphorin duplicity. Semaphorin is yet another mediator that's really important for our understanding of this paper today. In fact, it is the key player. It is the main actor. Okay. Semaphorins are really complex, but they're also, because of that, elegant and beautiful and super interesting in terms of how they can turn on and off a potentially pathogenic state to a healthy state. And you'll see how. Semaphorins and plexins act as ligand receptor pairs. Plexin is the receptor. Semaphorin initially is the ligand. Binds to its receptor, and then something downstream is going to occur. Okay, classical stuff. Where they exert pleiotropic effects in angiogenesis, tumor growth, oh, that's not good, bone homeostasis, that is good, remineralization, and of course immune activity, okay, activation. That's all what's happening with a semaphorin. Now, specifically, oh, by the way, there are multiple isoforms of semaphorins. You'll see them in a minute. Very complex protein structures. Not so complex we can't understand them, but more complex than you might initially think, first blush. Specifically, semaphorin 6D, which is the one we're going to talk about today, is a class 6 transmembrane type protein, and it functions as a ligand through association with its receptor. Only one plexin involved here, plexin A1, multiple plexin isoforms as well, of course. A phenomenon when SEMA 6D binds to plexin A1 at the membrane is called forward signaling. That just means that whatever's going to happen here, angiogenesis, remineralization, immune activation, all that stuff, that's called forward signaling. In fact, it's going to be functioning in the direction of mTOR initially. But SEMA6D also acts as a receptor. So not only is it a ligand, it's a receptor where the cytoplasmic domain or tail of SEMA6D, buried in the cytoplasm of the cell that's being activated, associates with another protein, which is got an interesting history, which is CABL-1. CABL is going to be a kinase that's going to be involved in all kinds of interesting things in the cell. In fact, one of its main pathophysiological associations is with chronic myelocytic leukemia, CML. We'll find out later how that all comes together in the research experimentation part of the paper. Oh, yeah. Now, like I always do in these lectures, I'm going to go back now and show you. Here's a paper published in 2011, December. Just Feb's Letters is the name of the journal, showing you the different semaphorins. Fortunately, we're only going to talk about semaphorin 6D. That's this character here. It looks like it's actually the most complex, which is just fine with us, right? Semaphorin 3A, I might just say as an aside, is the only secreted semaphorin. So this protein tends to be secreted and can move around from cell to cell. All the rest of them are basically immune responsive, and they're all membrane bound, okay? So these are membrane type semaphorins, 4D, 4A, 6D, and 7A, the ones that are most classically been examined. Let's look at 6D. It has a lymphoid interaction arm, and it has a non-lymphoid interaction arm, and that means it's going to recruit and associate with all of these other membrane type proteins that are going to then turn on. These are the plexins, you see, plexin A1. These are plexin A1 co-symmetries with all kinds of different domains in them. And these domains, of course, are shared across other proteins. Split GAP domain, GTPase binding domain, all these different domains are in these complex associated proteins in the plexin receptor. When that whole thing is activated by binding of ligand to the plexin A1, you get for example, in the immune response, activation of dendritic cells, really important as antigen presenting cells. I'm talking to T cells. Another intense topic from VerEvMed from lectures past. It also involves the osteoclast development. Again, I talked about that in previous lectures. But the big claim to fame here with SEMA6D, where you find most of it in the literature, is in the cardiovascular literature. It's actually involved in cardiac morphogenesis, both pathophysiological and physiological healthy responses. Okay, so that's all you really need to know. Membrane-associated protein is distinct from all these other semaphorins, and it links up with plexin A1 in its forward activation mode, right? All right. Now, the axis of this study, they're going to ask the question, perhaps an MTOR, SEMA6D, PPAR gamma, mediators I've already talked to you about now, signaling axis mediates the anti-inflammatory macrophage polarization via regulation of fatty acid uptake and metabolic reprogramming. That makes sense because PPAR gamma, that's what it does downstream. Transcriptionally activates that change in polarity from glycolysis to fatty acid oxidation. The SEMA6D looks like it's perfectly poised to be in the middle of that, right? Interacts with mTOR, and it also seems to be involved in PPAR gamma, but how? So that is, how does the transcriptional control occur? Which is the precursor and which is the product of that precursor interaction? And so you need to know what the sequence of events is. And this paper is going to tease that all out, particularly in macrophage polarity. Of course, you can tease this out in other systems, right? Semaphorins are not just in macrophages. Now, this research is going to suggest that the SEMA6D reverse signaling, that's the cytoplasmic tail functioning in the opposite polarity of what the ligand binding to plexin A1 initially did, which was anabolic. The reverse signaling plays a critical role in the control of macrophage polarization, which kind of is at least logically linear, right? Not trying to smuggle the conclusions into the premises, and I'm not doing that. I'm just saying it is logical that if you have a protein that's embedded in this axis, and it's an axis so things can swing on an axis, right? Then it makes sense that SEMA6D would be right in the middle, and indeed it is in the middle. So it seems like maybe it could play a critical role in controlling that polarization, and that's what we're going to find out. Now, a very simplified version of what you're going to hopefully have in your mind as I talk. You've got, remember we talked about Tolllike receptors. When I say remember, that was just in previous talks. We were talking about inflammatory bowel disease, and that's why I'm bringing it up here again. This comes from this particular hyperlink from Eureka Alert. I just want to show you that, for example, a cytokine receptor working through a Toll-like receptor can turn on mTOR, okay, it's activation mode. It turns on two other transcription factors, PGC1 alpha, which I talked about many times, YY1, which can actually induce mitochondrial biogenesis. You need a lot of mitochondria in activated cells because you need a lot of energy. Mitochondria are going to carry out electron transport chain oxidate phosphorylation. Now, plexin A4 system, right, the SEMA6D, binding to the plexin A4 system that we looked at in more detail in the previous slide, it's going to associate with that protein I told you, the kinase called CABL. It's going to be ABL-1. That complex, the SEMA6D, the cytoplasmic tail of that, now acting as a ligand, interacting with ABL, which again is a protein with a lot of history in diseases, it's going to turn on PPAR gamma. See, this is now we're going on a different pathway here. What PPAR gamma is going to do is transcriptionally activate CD36, which is going to allow the LDL and VLDL, very low density lipoprotein, low density lipoprotein, laden triacylglycerol, to be taken into that receptor, that orphan receptor. Lipoprotein lipase is going to work on it, making fatty acid from the triacylglycerol, and that is going to be used for fatty acid oxidation. So now you're switching from a glycolytic pathway, not shown here, to a fatty acid metabolism pathway, and you're going to get the alternatively activated state of the M2 macrophage. So this is the way it works. You see, so mTOR is working, it's doing double duty. It's making a lot of mitochondria. It's being anabolic. Oh, yeah, right? But it's also, if these are in place, if SEMA6D is in place and CLBL is in place, then it can induce the transcription of PPAR gamma, which is now going to turn on not glucose utilization, but fatty acid utilization. It's going to actually then cause this switch, right? It's going to make the whole thing turn and go the other way. And not only is it going to use different fuel, the best fuel, by the way, fatty acids, as opposed to boring old glucose, but also it's going to be able to turn on all the kinds of changes in the cell that are necessary to become anti-inflammatory. All right, now here's the first set of data. Do not be daunted. Okay, let's take a look at this. Note, this is a scatter plot here. And just like with all scatter plots, you get twofold upregulation when you see red. When you see red, think increases in gene expression. Then you see the blue, which is neutral, no change in gene expression. You see the green, and this kind of scatter plot, right? This this plot that's associated by looking at, it's like a temperature plot, right? Hot versus cool, right? At any rate, what's happening here is that the red of the genes turned on, green of the genes turned off. Okay, now treatment of macrophages with a mTORC1, actually it blocks both mTORC1 and mTORC2, a compound called taurin-1, rather than using rapamycin, we use taurin. All right, it's a selective ATP competitive inhibitor. It's nice to use competitive inhibitors because you can wash them right out with ATP, rather than using an inhibitor which is like suicide inhibitor. Like rapamycin does, right? Can't wash that out with a competitive because it's not competitive. All right, now, as a result of defective, when you add taurin and you screw up mTORC activity, you get defective anti-inflammatory, and by the way, you get enhanced inflammatory macrophage polarization. Whoa, okay, so that's what this is trying to tell us here. Now, if you look at the comparative transcriptome, which is what these data are all going to show you, it reveals that the expression of the anti-inflammatory signature genes, which is exemplified by this, resistin-like molecule alpha, which is called retin-LA, the araginase, remember, that's the urea cycle enzyme, the mannose receptor, don't worry about that, that's like a Tolllike receptor, it's going to be dealing with foreign bacterial antigens, actually, and by IL-10, okay, which is going to be a key anti-inflammatory system. They were all profoundly reduced in the presence of taurin-1. Also, SEMA6D expression is markedly reduced in taurin-1 macrophages. Now let's take a look at this. So here's a SEMA6D and there's SEMA4D. Notice that taurin, as you increase the concentration, shuts down the expression of SEMA6D, okay? Has no effect on that other semaphorin, which is good. That means it's specific, differentially specific. Likewise here, this compound here, this PPSS50, okay? It's going to be functioning on another aspect of the pathway that shuts down semaphorin metabolism as you increase the amount of this inhibitor, okay? Notice it has no effect on semaphorin 4D, but it completely shuts down SEMA6D, okay? That's because this compound works through another receptor, which is collinear with mTORC, a receptor kinase system called LAMTOR. So you got mTOR and you got LAMTOR, okay? So this compound, this terribly PPS550, it attacks LAMTOR. So what does that mean? Let's go over here and look at this. mTOR and LAMTOR promote anti-inflammatory macrophage polarization usually, or that is typically, via the activation of something called the liver X receptor alpha, okay? That's LXR alpha, right? That controls lipid and cholesterol metabolism, by the way. The reason it's called liver is where it was first discovered and described. Now, there's an antagonist. That's that weird compound we told you. It's not weird. It just has a weird name. It suppressed SEMA6D expression even after IL-4 stimulation. Now, IL-4 is supposed to stimulate this system, okay? But it did not affect the expression of SEMA4D, and that's what we saw here, okay? Suggesting that SEMA6D might act downstream of TORC1 and the LXR macrophages, okay? Because if it's working downstream, it would mean that if you inhibit LXR, which is what you did here, then yeah, you got a decrease in the main player we're looking at, the semaphorin 6D. Now notice also that in this slide you see the wild type level of urea is a lot higher than the SEMA6D. That's a double knockout, okay? So that means that the whole urea cycle is also compromised. And remember, that's going to be kind of like trans-reflective to our understanding of the urea cycle being turned on, right? The urea cycle is turned on in the anti-inflammatory response, right? So you're seeing effects on metabolites, and that's important, okay? All right. Now, after IL-4 stimulation for 48 hours, the double knockout SEMA6D, so they did a double knockout. This is a myelogenic line. This is the bone-derived macrophage lineage, okay? Bone marrow-derived macrophages, BMDMs. Sorry, now I remember it. Exhibited substantially reduced expression of ARG1, chitinase 3-like 1, that's Chi3/1, retin NLA, I don't need to go through that again, I already told you, IL-10, and this is all determined by quantitative RT-PCR. So that's what you're looking at here, okay? Now, these would be anti-inflammatory markers. Remember, these are the ones that are expressed in the anti-inflammatory polarity. So look at here. Whoa, right? Whenever you look at the SEMA6D double knockout, okay, in these bone marrow derived macrophages, right? These are like kind of like naive cells, naive macrophage lineage. You get less of the anti-inflammatory, again, less of IL-10, which is the transcription level, it's RNA. ARG1 gets decreased as well. And likewise, here's this chitin system, right? This chitinase 3-like, again, that's just another anti-inflammatory marker. But whoa, okay, so see all this, all of these are knocked down. So you look now at that REL protein. This is the protein level. These are transcript levels. And there's the wild type, okay? You turn on IL-4, you get more of it. That's what's supposed to happen. You're inducing the polarity in the wild type macrophages, these BMDMs. But when you have the double knockout BMDM, sorry, you can add interleukin 4 until you're blue in the face and you don't get really any increase in expression. That shows you've targeted the anti-inflammatory response by knocking out the semaphorin 6D, right? It's a case in point. All right, now take a look at these pro-inflammatory cytokines. These are again at the RNA level. There's tumor necrosis factor RNA. There's interleukin 6 RNA. There's also the inducible nitric oxide synthase 2 messenger RNA, which is also associated with the pro-inflammatory response. Okay, all of these are jacked up in the Semaphore double knockout, right? Okay, so and here is again the interleukin 10. That's the anti-inflammatory. Again, just proving to you that there isn't something funny with this data set. That is again turned down in the double knockout. Pro-inflammatory cytokines and the inducible nitric oxide synthase, which is another pleiotropic associated enzyme with pro-inflammatory, all up in the double knockout, but the anti-inflammatory down. That's just showing you all. This is all transcriptional levels. It's RNA, QTR, PCR, RT-PCR. Now, okay, I think I just said all that. Additionally, urea production, which is an indicator of arginase 1 activity, is markedly reduced. Okay. Now, under LPS plus interferon gamma stimulation, this is how you activate a macrophage. The SEMA6D double knockout bone marrow derived macrophages, the naive macrophage, express substantially higher levels of this IL-12P40. I just went through all this. Remember, TNF, IL-6, and inducible oxide synthase, NOS2, right? And then the wild type, whereas the levels of IL-10, which is anti-inflammatory, markedly reduced. I just went through this. Okay. Consistent with this, double knockout, SEMA6, SEMA6 negative double knockout, BMDMs produce more pro-inflammatory cytokines. I just showed you that. See? Okay. See, there it is again, transcriptional level. Okay. Actually, this is protein now. This is IL-12. Okay. You get an increase in the knockout. You see, here's tumor necrosis factor, you get an increase in the knockout, even though you're inducing, this is stimulation with LPS and interferon gamma. You're getting more and more of the pro-inflammatory cytokine, but you're not getting any of that anti-inflammatory cytokine. So the polarity is unable to switch because of the double knockout, which links Semaphore and 6D right in the middle of that axis, as was predicted. Okay, more data. Expression of the most anti-inflammatory related genes profoundly lower in the double knockout. This is in peritoneal macrophages than in wild-type peritoneal macrophages. So this is what this data is looking at. These are peritoneal macrophages taken from mice, okay? So if you intraperitoneally inject IL-4 antibody complex, this is supposed to induce the system, right, into the wild-type or the SEMA6D double knockout to trigger this polarity switch to anti-inflammatory, right? You don't get it. It doesn't happen. We observed a reduction in the expression of anti-inflammatory signatures in the SEMA6D peritoneal macrophages in comparison with the wild type, okay? So you take a look here. This is how you add chitin, which is an activator of the activation of the macrophages to be pro-inflammatory. And you can see here that all of these anti-inflammatory, right? All these anti-inflammatory markers are not tuned up here. They're not tuned up. That's the only one that is, okay? All the rest of these are not tuned up upon induction of the transcriptional level. When you add interleukin 4, you're supposed to get a boost in all of those. You're supposed to get a boost in the anti-inflammatory because IL-4 is supposed to cause the triggering of the change in polarity. Hey, look, it's really bad. This is a log scale here, a tremendously large log scale. That only goes up to 1.52. There's 400, right? IL-4 really gives you that boost. That's why they used it. Oh my gosh, none of these are going to be turned on when you have the double knockout. That means semaphorin is absolutely critical for that change in polarity, okay? It's just showing you survival rate. What happens when you're using LPS is saying, would it just kill cells, okay? Well, the wild type cells weren't as killed as badly as the double knockouts. And of course, that kind of makes sense because in the double knockout, right, you're starting to destroy the tissue, right? You're starting to destroy the tissue. But under low LPS induction, you get less of that, which makes sense. It's LPS associated, LPS dosage dependent, right? All right. Again, what you're looking at here, this is just protein, right? This is the pro-inflammatory, more of it in the double knockout, more of the double knockout every time, except in the anti-inflammatory interleukin 10, where you get a tankage of the protein level when you have the double knockout, okay? I want you to remember that typically the tolerogenic, that's the polarity you want that's anti-inflammatory, tolerogenic macrophage polarity, would have expressed IL-4, 10, TGF-beta, IL-13. And none of that stuff is going to go down here because you're blocking that system. Now, here we go with a summary till now. Semaphorin can act as a ligand, or its cytoplasmic tail can perform as a receptor. Okay, that's in the literature, and that looks like that might be what's going on here, as you'll see in a moment. Anti-inflammation gene expression is paradoxically reduced by blockading MTOR, and semaphorin 6D is likewise suppressed. So that makes sense. It's not really paradoxical because you're decreasing the transcription of semaphorin 6D, which cytoplasmic tail is required for flipping the polarity. So it's not really paradoxical. It's pseudo-paradoxical, right? SEMA6D double knockout mice are unable, upon chitin stimulation, to generate anti-inflammatory macrophage. We just saw that. So you got low expression of the anti-inflammatory cytokines, right? Once we were talking about, and rather become militantly polarized as pro-inflammatory, right? They become even triggered the wrong way when you get to double knockout, when you use a double knockout. This is further demonstrated by attempting to induce with IL-4. You should really get a boost, right, in the anti-inflammatory. The wild type, you can turn that on. Even after you've stimulated with chitin, you get none of that in that double knockout. So this has got to be downstream from that interleukin-4 receptor, which makes sense. Looks to be, this so far, an amphibolic. That means it's localized within the cell in different times and different locations. Multi-protein complex, which we saw already some cartoons of. Switching mechanism, whereby mTOR stimulation emblematically initiates anabolism and also the pro-inflammatory response here, but also is the efficient, that is the direct cause of a semaphorin 6D synthesis, which that is the transcription of it, which ultimately turns the activated macrophage into the fatty acid translocator and beta oxidizing anti-inflammatory polar existentiale. Don't worry about that, I'll mention in a minute. Essentially, an elaborate intracellular fail-safe feedback inhibition mechanism. That's what we're looking at here. It's intracellular, and it's not global within the whole organism, like, say, endocrine system works. Now, I call this an existentiale because what that means, this is a philosophical term from Martin Heidegger, actually. Existentiale means it's understood as existing as an individual with discrete predicates, right? With discrete predicates, with discrete properties. And isn't that exactly what we've done to the semaphorin? We've created a polar existentiale, right? So I like using words like this because it's much more precise than just saying a different form. It's a different existentiale. That means it's existing as, in real time, so it's an event, an individual protein molecule and protein complex system with discrete predicates, which are unique because of the change inculcated and induced by using the whole mTOR system. Yeah, okay. Now, you can't express PPAR gamma in double knockout SEMA6D mice unless you supplement with IL-4. That's what this is showing you. It's PPAR gamma, okay? It's wild type versus SEMA-D, and even with IL-4 here, okay, at the RNA level, you're not getting any activity, right? Only when you're looking at relative amounts of PPAR gamma messenger RNA, you get non-significance when you further induce with IL-4. So in the presence of IL-4, you get no activity, but even when you superinduce, okay? Looking at PPAR gamma, so it's showing that it's required. Take a look at this. IL-4, when you add IL-4 to the wild type, that's when you get PPAR gamma. When you have the double knockout, not so much. This is a protein blot, a Western blot, okay? Now, there's PPAR delta. Any effect? No, okay? So it's showing you, again, differential control over expression of PPAR gamma as related to interleukin-4 induction, okay? Now, likewise, take a look at these cells. Oxygen consumption rate, that's over here, OCR, in picomole per minute, that's the y-axis, is reduced in the double knockout mice. So that means you have impaired what? When you're not consuming oxygen, what are you switching to? Remember, if you can switch, beta oxidation, you use a lot more oxygen. Beta oxidation of fatty acids is a oxygen requiring system, okay, unlike aerobic glycolysis. And so you get less oxygen consumption because you're getting less beta oxidation in the knockout. So there's a good physiological phenotype readout. I like the fact that they did that. Now, poor expression here, we're looking at CD36 messenger RNA, and remember, that's the orphan receptor that takes up the fatty acids from the triacylglycerol, from the circulating LDL and VLDL via the acyl hydrolase. And fatty acid binding protein, shown here, these are transcriptional levels in the double knockout mice, and even in interleukin-4 no longer rescues it. So that means that the expression of these, even when you induce with interleukin-4, you get no rescue. So that tells you that in the when you have the double knockout, and now you're looking at the transcriptional activation that is required by PPAR gamma. PPAR gamma is now the player that controls this. PPAR gamma is controlled by interleukin-4 and wild type. But look at here, no way do you get anything downstream from PPAR gamma because you don't have any PPAR gamma. So you can add interleukin-4 until you're blue in the face, and you are not going to get an induction of these genes, which would normally be induced by PPAR gamma, because it's not there, right? There's not enough of it. So you don't get the induction like you do in the wild type. Okay, right. Now, you need interleukin-4 stimulation, but not plexin A4 receptor for SEMA6D to obtain that expression. So, so that's interesting. The plexin A4 was needed initially, but no longer is it necessary. So with or without plexin A4, right, even with a high dosage of it, right, the PPAR gamma is just on as long as you have interleukin-4. Right? So that means plexin 4 is no longer controlling this. It's no longer, so that again means it's somewhere downstream. However, you do need interleukin 6 or tumor necrosis factor to rescue the double knockout mice, okay? So when you add interleukin 6, right, you're able to rescue the phenotype, okay? You're able to rescue the phenotype for tumor necrosis factor and for the induction of IL-6 itself, right? When you are downstream from that plexin receptor modification. So what is interleukin 6? This is a direct quote from Cold Spring Harbor Perspectives in Biology 2014 paper. IL-6 promptly and transiently, see I'm saying it in a different voice, you know this is a quote, produced in response to infections and tissue injuries, contributes to host defense through the stimulation of acute phase responses, hematopoiesis, and immune reactions. Although its expression is strictly controlled by transcriptional and post-transcriptional mechanisms, dysregulated continual synthesis of IL-6 is actually very pathophysiological. In fact, you see it in chronic inflammatory diseases, such as those associated with obesity, by the way, and in autoimmune diseases, such as what happens in IBD, Crohn's, ulcerative colitis, things like that. Okay? All right. So that's the point here. IL-6 is going to turn this on, and it will rescue. So that means it's way downstream. All right. So you need IL-6 or tumor necrosis factor to rescue the double knockout mice. Now, inhibitors of ABL1, remember now we're bringing this fellow back. Now this dasatinib, okay, is actually an inhibitor of the SERK kinase. And the SERK kinase intracellularly cooperates with ABL1. Okay, so they're using dasatinib and increasing the concentration. And look what's going on to show that ABL1 is important. You had dasatinib, which is going to inhibit SERK, which works with ABL1, and you get less arginase transcription. You get less PPAR gamma transcription. It's funny they put the G there and not the gamma sign. I guess something happened to the computer software. And this is just showing you all these different genes for the anti-inflammatory cytokines, the IL-10, the retin-1A. I hate the fact that it looks like retinal. You know, I was really confused when I first saw this. Not, of course, you know that's resistant. There's the PPAR gamma, much less than the wild type, right? There's the ABL1. This is showing you not using dasatinib. This is using an interfering RNA. So showing you the same phenotype, right? You interfere with ABL1, you get less pro-inflammatory cytokines. Isn't that interesting? This is just showing you that the siRNA works. You get less of that transcript, right? You get less of that protein too. Right? Beta-actins are control, loading control, as well as a control over activity of the siRNA, both here. Sometimes that's not good to do that experimentally, but I think it's okay here. I have to think about it, but I think it's okay there. Now, here's the dasatinib, increasing the concentration. We already showed you. Wow, you just get rid of PPAR gamma. Okay, it's gone, right? And then there's that REL M alpha. That's just a subunit of this protein here. Again, showing you that it goes away with the dasatinib, even in the presence of IL-4. Okay, IL-4 is not going to be able to rescue this. There's the beta-actin loading control. So, inhibitors of ABL1 will annihilate anti-inflammatory cytokine production, suggesting that ABL1 cooperates with the SEMA6D, obviously, to induce anti-inflammatory polarity, and that works through PPAR gamma. That's what this data shows us. ABL1 is, what is it? It's a non-receptor tyrosine protein kinase that plays a role in cell growth and survival. It's actually inhibited by AB1, okay, whose activity is controlled by ABL1 itself through tyrosine phosphorylation. So the protein autophosphorylates, but it's also auto-inhibited by the product. Wow, Very cool, right? That's because it's so lethal. It's such a dangerous protein. It has to be controlled at multiple levels. Right? And it's all at the level of tyrosine phosphorylation, autophosphorylation. Also inhibited by imatinib mesylate, also known as Gleevec, which is, of course, a big CML drug, although now there's a lot of resistance to it. Gleevec was one of the first drugs that was actually designed to be site-specific for a particular protein in leukemia. And in fact, the people that came up with that were from the University of Washington, 2 researchers there. They got patented, made quite a bit of money on that. UW made a lot of money on that, but now there's getting to be resistance to it. Like every drug, there's resistance. Anyways, used for the treatment of chronic myeloid leukemia, as I told you. Dasatinib is sold as a compound, a pharmaceutical called Spiacel from Bristol Myers, and I told you that inhibits the SERT kinase. And that's why it's used up here, okay? Which cooperates with the ABL. It's another actually drug that's involved in inhibiting CML. So remember that the way that you get CML is because of something called the Philadelphia chromosome, not because it was made in Philadelphia, because it was first described in a lab in Philadelphia. Okay, I know, terrible the way these things are named. I bet a geneticist had something to do with that. Come on, chromosome, you bet. Geneticists always give funky names. So you got the BCR ABL1 fusion gene. That's called the Philadelphia chromosome, and that's in CML. It's the efficient cause of CML in humans, where BCR is actually a serine tyrosine kinase. Okay, we're unmasking what it does. And it's also a guanylate nucleotide exchange factor. So that's what BCR does. So when you link that to this other kinase, you get tremendous leukemia when you have that happening inside a leukemic cell. That's how it happens. All right. And that's why Gleevec was made, and that's why Gleevec is becoming less sensitive, because it's a target, like any other drug. Now, more data. PPAR gamma is a downstream regulator of SEMA6D. So the SEMA6D control over PPAR gamma transcription governs macrophage polarity. So take a look. There's IL-10. Again, when you have the double knockout, less. There is interferon gamma, which is, of course, pro-inflammatory. The double knockout, you get more, a lot more than the wild type. Another pro-inflammatory, this one's a really, really nasty pro-inflammatory, IL-1 beta. That's like the mother of all nasty ones, okay? Again, IL-17A, another really potent pro-inflammatory cytokine, jacked up in the SEMA6D double knockout. Tumor necrosis factor, jacked up. And there's PPAR gamma, just to show you, this is something weird with the data, tanked in the double knockout. So pro-inflammatory cytokines are upregulated and anti-inflammatory cytokines are downregulated in that double knockout SEMA6D mice. And these animals are predisposed, this is important, to colitis. Why? You get colitis from having a high level of chronic inflammation. You would get chronic inflammation in these double knockout mice because you don't have a way of switching the polarity of the macrophages to the anti-inflammatory response. Simple QED. All right, here are the concluding remarks. SEMA6D-induced PPAR gamma transcription regulates fatty acid uptake and oxidation in the anti-inflammatory polarized macrophage. That's clear from this data. SEMA6D transcription itself is down-regulated when you inhibit TORC. Again, proven in this paper. SEMA6D downregulation, then, when you get it, results in the impairment of the anti-inflammatory macrophage polarity while promoting the inflammatory macrophage lineage. The absence of SEMA6D, which we have with the double knockout in several of these experiments, reduced the expression of PPAR gamma and its target genes, CD36, FABP4, thus blocking the switch to fatty acid oxidation from aerobic glycolysis. Remember, the PPAR gamma transcriptionally controls beta oxidation, mitochondrial biogenesis, and also all the proteins necessary to take a fatty acid and turn it into acetyl-CoA, and thus making NADH and FADH2, which can be run through the ETC oxphos to make a lot of ATP. SEMA6D functions as a supramolecular axis. These are my words here. That paradoxically, or maybe pseudo-paradoxically, or maybe actually paradoxically, both maybe, links mTOR with PPAR gamma instantiating macrophage polarization. That's all going down via its 389 amino acid receptor functional cytoplasmic tail. Okay? Now, we know that because there's a tyrosine mutant, a variant of SEMA6D, which is going to lack that linkage ability. Once you have that mutant, you have that tyrosine mutation, you can't do any work, you can't interact protein-protein with CLBL. That could not, that using that protein, you can't, when you overexpress it in the double knockout, semaphorin double knockout, can't rescue the anti-inflammatory macrophage polarization in that knockout, okay, in those macrophages. So that tells you that you need that association with ABL, which again links it to this 389 amino acid receptor functional cytoplasmic tail. I think, again, QED, right? SEMA6D has a proline-rich region, just to let you know some details, for the SH3 domain binding site in that cytoplasmic region, which apparently is important for the association with CLB kinase, ultimately inducing the transcription of PPAR gamma, thereby promoting the anti-inflammatory property in those macrophage lineages. In the immune system, CLBL is negatively regulated by the transcription factor NF-kappa B. This is normally what happens, which is a housekeeping signaling system whereby NF-kappa B is a first responder to things like inducers of inflammation, reactive oxygen, TNF-alpha, circulating IL-1 beta, and of course, the bacterial antigen lipopolysaccharide. So that's how normally it's controlled. CLBL is negatively regulated. So all this wouldn't happen if you get less CLBL made. Right? So this is all beautifully controlled. No paradox, really. What is our diabettological perspective, you might ask? Dr. Guerra certainly asks. Simple canonical firing of mTOR, sorry I screwed up a little bit on the way those are capitalized, mediated anabolic promotion is as mistaken, if you think that's all it is, as the naive assumption concerning endocrine hormones. Systems aren't simply on or off or unidirectional. Oh, now you have a whole lot of this hormone effect and that's all there is. You know, if you know anything about endocrinology, there are tremendously elegant feedback loops. You get the expression because of factor expression for, say, endocrine hormones, steroid hormones. At the same time, you start turning on the inhibition of the expression of the factors which will allow for the transcription of those hormones. That happens in the global homeostatic system between organs in the body. This is just showing you that it can happen intracellularly using the semaphorin complex and plexin and mTOR. Okay. And ultimately the PPAR gamma. So the mTOR SEMA6D, PPAR gamma reverse paradox demonstrates the necessity for a four-dimensional systems teleology for understanding intracellular phenomenon. What's the fourth dimension? Okay, you got the three normal spatial dimensions. The fourth dimension is time. This takes time. This is turned on, but not turned on in exactly the same time signature. If it was, it would just be what? Self-defeating. So this is turned on and it's set to go into motion so that later on the macrophages, pro-inflammatory, all of a sudden anti-inflammatory, you see? It's a beautiful system. It's like watching the water come up and then subside, and water come up and subside on a sandy beach, right? Leaving things behind each time, right? And taking things away. Always refining the substratum sands. It's nice and fine and particulate. Right? All right. Now, the environment, which in this system today is called as simply the signaling we were looking at, genetics, which we didn't go into detail, except for that tyrosine mutant, but you get inherited genetic markers via single nucleotide polymorphisms, copy number variations, etc. And the event epigenomic alteration, okay, of the various molecular interactions. Now, there's an epigenomic thing, wasn't in this paper, but I'll tell you in a minute about this. Of the various molecular interactions must all be studied and verified in each unique cellular system through time. I'm giving you an argument for studying this from a diabettological level. These people did a very good indentation in that way, okay? Don't need to do any more for this paper, but I'm just telling you, you have to think about event epigenomics. Okay, what do I mean by that? For example, even though not shown in this paper, of course I dug this out of the literature. Colorectal cancer, which is exactly what this paper is ultimately looking at in terms of biomedicine, right? Why do you get this pro-inflammatory IBD? Because you can't switch to the anti-inflammatory macrophages, right? Because of semaphorin problems or something with the signaling. Colorectal cancer is characterized by hypermethylation. Now, I talked about this in the previous talk, by the way, of an enzyme called hydroxyacyl-CoA dehydrogenase trifunctional multi-enzyme complex subunit beta, also known as HADHB. That enzyme complex catalyzed the last three steps of, beta oxidation of long-chain fatty acids. So colorectal cancer is characterized when you hypermethylate the cytosines in the promoter regions of that gene. When you hypermethylate, what happens? You get less expression. Right on. Now, both that enzyme, which is canonically characterized in CRC, okay, low levels of that, that's like a hallmark for having CRC. Both HADHB, that enzyme, and SEMA6D are both downregulated via epigenetic hypermethylation by studying the methylome versus the hydroxymethylome, which gives you an idea of how things are switching, because hydroxymethylome is on the way to not being methylated because it's a degradation of the methylation signature. I won't go into any more detail than say that for those of you that like that sort of stuff, like me. Anyways, via epigenetic hypermethylation in human colorectal carcinoma. And I got that from Clinical Epigenetics, a journal, and there it is, brand new publication again. Okay, there you go. So it looks like SEMA6D is under the control of methylation. And when you get hypermethylation, what happens? You don't get double knockout, but you get a suppression of expression. Suppression of expression. What happens? You don't get less transcription. And that is linked, okay? Linked to, as is the hypermethylation of this enzyme, colorectal cancer. Wow, if that isn't a good case in point, I don't know what it is. In a nice way to thread the needle. Now, just to remind all of you, here's my diaventome. You have the genome on one axis, the environment on the other, and then because this is triplanar, right, you have the immunoepigenome on the third axis. Okay, in the middle is the individual system. Okay, the individual system then is controlled by genome, environment, and the immunoepigenome. Now, the genome is inherited, imprinted gene expression patterns, single nucleotide polymorphisms, splice variations, as I said, copy number variations. If there are any inborn errors of metabolism, they're in your genome, right? Unless they've been corrected via recombination. The environment can be changing stress, both internal and external. External meaning like, say, changes in temperature or changes in pathogen invasion. So external can involve pathogens or xenobiotics. That's external. Diet is external. Medical treatments are external. Psychological stress starts to get into internal. But also neurological, cardiovascular, metabolic disease like diabetes. Signaling like shown in this paper. Wound healing, neoplasia, and cancer and aging. All of those are environmental factors. What is the immunoepigenome in this triplanar system? Real time, because it's immune effective, right? And because the epigenome is changing the expression within the immune cells themselves or in associated tissues like epithelia or even endothelia. Real-time response to and alterations of immune surveillance and activation, which we just saw here, inflammatory response in general, we just saw here, energy, like shutting down T cells, which I talked in previous VerEvMed lecture, polarization, again, we saw it here, immune memory, epigenetic writing, reading, and erasure. That's all part of this huge immunoepigenome, which is being talked to by the genome and the environment in a three-way interaction, ultimately leading to a specific self-organized expression, which we call the diaventome. So, what is diaventology? It's discussion of an adaptational processive accounting. So again, this isn't a substrate ontology. This isn't a substance ontology. This is an event ontology because it's through time. Every time I look at biochemistry, I have to see things as events, not as substances. Substances are static and they tell you really nothing. An adaptational processive accounting. Notice I didn't say progressive, processive accounting of the rational human mind through cellular molecular event ontology or this is the original conception, okay? So how does the mind function? Same way. Again, but via immune sculpting. Now, the immunoepigenomic induction of neural, which was the original conception I had, but also the endocrine and metabolic response to micro and macro environment through classical constitutive surveillance and acquired effector cellular and humoral defense stratagems using reversible covalent modification and hydrophobic interactions of nucleic acids, proteins, and lipids. Okay, that's all part of the package of what's going on intracellularly. That may become, diabettology, a central molecular theory for the kinds of things we talked about today, but also how you link free will-driven, agency-based existing individual adaptation, which ultimately leads to an epigenomic signature, and knowledge acquiring epistemology that explains the core event ontology of human existence. It's way out there, but not way out there intellectually. It's way out there like in terms of it needs a lot of research to be able to build this. That's what I'm promoting. So what are some events in physiology and pathophysiology, Dr. Guerra? Give us some. Molecular and cellular events that occur across mechanisms that result in real-time modification and changing conditions. Those mechanisms include, but of course aren't limited to, always put that caveat in, replication, recombination, repair of DNA. Those are dia events. Transcription, alternate splicing, translation of RNA. Signal transduction, like we talked about today. Membrane fluid dynamics, don't worry, that's coming up. Cell fate repertoire, like autophagy, apoptosis, senescence, necrosis. We've talked about many of those in previous lectures. Developmental programs, including embryogenesis, right? Cellular division and differentiation, including adipogenesis, that is, neurogenesis, myelocytogenesis, lymphocytogenesis, immune cell polarization today, gametogenesis, oncogenesis, on and on and on. Okay. So we're done today. That's me, Dr. Dan Guerra. That's the Great Divide in Colorado, a place I like to visit a lot. There's our email address. There's our website. We are scientists verifying published evidence in medical biosciences. My Facebook page. Look to that for all of these VerEvMed lectures. There's my individual email address. Contact me on any content you see. If you just want to interact with us, that's how you're going to be able to do it. So, without further ado, I want to say, like I always do when I say goodbye, is bye for now. I'm going to take off these glasses, which I had on a long time, and they're kind of giving me a headache, but aren't they fashionable? Anyways, you guys have a good weekend, and we'll be coming up soon, hopefully next time, with some membrane fluidity discussion in diabettology. Hope this was a provocative and interesting paper for you. I thought it was one of the better published papers in immunological journals for a while, because it included a lot of different aspects of what's going on intracellularly in terms of polarity, and I really liked the way it came together with MTOR. So bye for now.